Okay, Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, we finally reached the transcription topic under the chapter 4. We are about to end our Bio 150 soon. Okay, we still have one more topic which is on translation. But first, let's have a look at the transcription. Okay, so I can be reached through my email down there, fathiah@uitm.edu.my. So here I paste a chromosome photo, a chromosome diagram here. If you can zoom into the chromosome, you can see that they are basically comprised or being composed by two strands of DNA. You can see that we have the sense strand and also the anti-sense strand. Okay, you can see that where's the 5' and the T', and then let's have a look at the anti-sense strand. You have the AT, GAC, GG, AT, CAG, and blah blah blah. And the complement RNA strand, let's say if you have A at the anti-strands, then meaning that you will have a couple which is U. Okay, and that's the things that we're gonna learn during this transcription topic. Okay, now let's have a look at our 6 learning outcome. If I can refresh you back, number 1 is to describe the basic principle and process of mitosis and meiosis. Number 2 is to differentiate between mitosis and meiosis. Number 3 is to differentiate between the 3 DNA replication models. And number 4 is to explain the semi-conservative of DNA replication. And number 5 is to define the function of mRNA, tRNA, and ribosome. So my part is the last one, which is to describe the process of protein synthesis. I have 10 fun facts to share with you guys. Okay, let's together look at this. Number 1, if you unwrap the entire DNA that you have in all your cells, you could reach the moon about 6,000 times. If the entire DNA in your body was put end to end, you can also reach the sun, go and back over 600 times. And there are about 100 trillion cells in your body with 3 billion base pairs in the DNA code within each cell, and over 99% of our DNA sequence is the same as other human. So you are similar to next human that are sitting beside you, about 99%. It takes about 8 hours for one of your cells to completely copy its DNA, and it would take about 57 years to recite your ATCG at a rate of 100 base per minute if you started tomorrow morning. 57 years, guys. Our genes are very remarkable. Okay, we are sharing 98% of our gene with chimpanzee. We share 90% with mice, 85% with zebrafish, and 21% with worm, and not to forget 7% with a simple bacteria such as E. coli. And 1 out of every 180 children is born with a chromosomal abnormality. Later on, if you decide to learn on genetic, then you will go further about this one. And the last point here, we still don't know the function of over 80% of human DNA. So biology is basically a very broad field to explore. Okay, first of all, the transcription process is, this process is basically take place in the nucleus. Why? Why I say so? Because it involves DNA. You know that DNA is contained in the nucleus, so it take place in the nucleus. So it is the process of making mRNA from a DNA template. Remember this, transcription is about making mRNA from a DNA template. It is different with the replication. Replication is the process where you produce DNA from a DNA also. Okay, so these are very similar to DNA replication. So, but then from this transcription, when you produce RNA, RNA will then leave the nucleus and go to the ribosome, where in the cytoplasm. You know that ribosome is not located in the nucleus, they're located in the cytoplasm. So there soon you will have a translation process. Okay. So as I told you before, for transcription we use DNA as a template to produce mRNA. So here we can say that from DNA they are transcribed into mRNA. So they are very similar to DNA replication, and just remember, as in replication, addition of a new nucleotide that occurs at the 3' end of a strand. Okay. And have a look at the photo at your right side there. We can see that, let's say we have a DNA molecule, a double-strand DNA molecule. Let's say the green colored one transcribing the gene 1, and the yellow colored one is transcribing the gene 2, and the blue colored is transcribing the gene 3. So you have a 3 type of gene. So down there also we have a DNA strand transcripted into RNA and translated into polypeptide. So this part is the middle part, which is where the DNA strand will produce the mRNA. That's what we call as transcription. And you can clearly see that, let's say we have a DNA strand there. If we have the base A, C, C, G, G, C, A, when they transcripted into RNA, the A that previously paired with thymine, adenine with thymine in the replication, will be replaced with the uracil. So there will be no more T, that will be replaced with the U. So A will couple with U, C will couple with G, similar as replication, and again at the end A couple with U. And from that RNA strand, the 3 base pair, we will read these 3 RNA here as a codon. So U is a single codon and will be translated into a type of amino acid. Okay, pairing in transcription is just as similar as in DNA in terms of hydrogen bonding, where A will bind with U with 2 hydrogen bond and G will bind with C with 3 hydrogen bond. Okay, at your right side there, you can see that they are anti-parallel to each other, meaning that if you have at the left is starting with the 5' end, so the partner at the side of that 5' end should be the 3' end. Okay, so you can see that the A bind with the T with 2 hydrogen bonding, and below than that you have the C with 3 hydrogen bond, they bind with the G. So these 2 different hydrogen bonding here give a curl and give a shape to the DNA structure. So the RNA is made so that its sequence is complementary to the DNA, whereby A transcribe U, G transcribe C, T transcribe A, and C transcribe G. The binding between the new forming mRNA with the DNA is using some sort of complementary coding, where if you can see that if we have the template DNA is ATG, CTT, AAC, CGG, and TT. So the transcribed mRNA must be UAC, GAA, UUG, GCC, and AA. You can see that A complement with, or A transcribe U, T transcribe A, G transcribe C, and C transcribe G. Okay, your right side is basically is simplified of the transcription, whereby you have 2 strands there. You have the DNA helix, which is double strand of DNA. There comes the RNA polymerase. Okay, when there comes the RNA polymerase, they will open up the DNA double helix. When they open that double helix, so the elongation, which is the light blue colored one, they are producing a new strand of RNA. So that process there is the elongation until they find the stop codon, whereby they have to stop the process. So we will go one by one. So we have 3 main steps in transcription. We start with initiation, number 2 is elongation, and number 3 is termination. Okay, so now just imagine, if you have double strand DNA, now we are about to open up the double strand DNA so that one of the strand can be the template for a new mRNA strand. Okay, so this transcription is basically catalyzed by the enzyme named as RNA polymerase. So you can see that from the photo there, down there you have the RNA polymerase 2. Okay, but first of all, RNA polymerase binds and unwinds the DNA strand at promoter region. So what is the promoter region? It is a sequence of TATAAA, or normally known as data box in DNA. So just imagine, if you have a double strand DNA, then you found that they have a Tata box sequence, meaning that it is a sequence for promoter. Okay, so these are the promoter region. So later on, the RNA polymerase will come and bind to that promoter region. Okay, so transcription of gene began when the RNA polymerase can find the promoter region or the Tata box. But then the promoter region itself or the promoter sequence is not transcribed. Okay, you can see that we have the Tata box. It is a promoter region. Here comes few transcription factors. Then when you have the transcription factors, then only you have the RNA polymerase 2. Then only that kind of enzyme, so remember the one that you learned in your chapter 1, RNA polymerase 2 will come and recognize the region so that they know, okay, so this is my starting point, I have to start here. Okay, then they will move forward, okay, towards the process. Okay, now we have initiated the transcription process. Now, meaning that we already have the RNA polymerase. Okay, now before we go further, let's have a look at the inside of the RNA polymerase. Okay, you can see that you have a a double strand DNA been opened up. You have two blue colored strands there. Okay, the upper one is ATC, ATC, CAA. Alright, and the lower part is the TAGGTTAA. So now we just have to pay more attention on the template strand of DNA. So why does we call this strand of DNA as a template? Because you are using this DNA strand as a template to produce a new mRNA strand. Okay, if you look at the non-template strand, nothing's happened there. Okay, so now we are using the template strand of DNA to produce the new mRNA. You can see that, let's say the first nucleotide there, C, the couple should be the G. A with T, meaning that the T at the template strand are attracting the nucleotide with A. Okay, the A at the template strand attracting U to come, G attracting C to come, G attracting C, and T attracting A. So here come the RNA nucleotide. They come pieces by pieces. Okay, you can see that they are forming the new strand at the end of the 3'. So the polymerase basically, when they move downstream, they are unwinding the DNA together with elongating the RNA transcript from the 5' direction 5' to 3'. So 5' to 3' here is referring to the new strand of mRNA. So RNA polymerase transcribe and add complementary bases on the gene region. After the RNA polymerase move away from that strand, the DNA strand that previously have been separated will be reformed a double helix again. Okay, the process of elongation will continue until the RNA polymerase encounters a stop sequence. At this point, transcription will stop and the RNA polymerase will release the DNA template so that DNA template will bind again and form the double helix DNA. So RNA polymerase transcribe the terminator sequence on DNA, and at this moment all the components will separate. mRNA will be released and the RNA polymerase will detach from the DNA. So the terminator sequence, which codes for polyadenylation signal of 5' AAU AAA 3' in the mRNA, causes RNA polymerase detach from DNA. This sequence here we call as a poly-A signal sequence because they have more A, meaning that they are starting, if the RNA polymerase walk at the transcripting the DNA strand, at the moment they created, or at the moment that this poly-A, they encountered this poly-A, then the RNA polymerase will detach and the process of transcription will be terminated. Okay, so basically we have completed the process of the transcription where DNA transcribed into mRNA. So the transcription process is happen in the nucleus. You can see that because transcription is about processing mRNA and they are using DNA template. So DNA can only be found in the nucleus. So this transcription process is happening in the nucleus. Later then, mRNA will be translated into protein. So these are the translation process. So that translation process has to happen outside of the nucleus because you need to use the ribosome. So mRNA will move from the nucleus through the pores in nuclear envelope and to the cytoplasm where they can find the ribosome and they proceed with the translation process. The mRNA which has been transcribed up to this point is referred to as pre-mRNA. So the processing must occur to convert this into mature mRNA. What processing? The RNA processing. Okay, so the processing to mature the mRNA have to involve the capping, the polyadenylation or tailing, and also the splicing. Okay, the three processes to mature the mRNA is capping, polyadenylation or tailing, and splicing. So what is capping? Capping is basically you put something. Okay, so here you are, you do some addition of a methylated guanine. Cap confers protection. They give protection to the mRNA, and this is necessary as RNA is much more unstable than DNA. This capping occurs at the 5' end of the mRNA transcript to protect the mRNA from degradation. For polyadenylation or tailing, it is an addition of a poly-A tail to confer also protection to the mRNA, and several adenine will be added. Basically, about 200 base of adenine will be added to become a tail to the transcript by the enzyme poly-A polymerase, and it occurs at the 3' end, protecting the mRNA from degradation. So bear in mind, you have the capping and you have the tailing. Capping is by putting some methyl guanine at the cap at the 5' end, and tailing is putting some poly-A tail at the 3' end. Splicing, what is splicing? It allows one genetic sequence to code for different protein. So this conserves genetic material. So here you have for the mRNA immature one, you have the intron and the exon. So what is introns? Introns is a non-coding sequences that are removed, will be removed, and the exons is the coding sequence. They are joined together by ligation to allow many proteins to be made from a single pre-mRNA. Okay, so this is what I mean with splicing. You can see at the pre-mRNA level, you still have the exon, intron, and the exon. Okay, after the splicing part, you will definitely produce a mature mRNA without the intron. All you have is the capping, 5' cap. You have the protein coding region that coming from the exon, and you have the poly-A tail. So by the end of the transcription, mature mRNA has been made, and this acts as the messaging system to allow translation and protein synthesis to occur. The basic question for this transcription is, can you describe the process of transcription? Can you name me the three main processes for the transcription? If you can answer that question, then you already mastered this transcription. Bye!